In this video, we will be going to the second part of experimental technique 2, alternative to practical, separation technique 1. There are many separation techniques, and we will be starting with separation technique 1, which is paper chromatography. You know, there are many criteria that can be used to determine the purity of a substance in a mixture. There is a boiling point, there is a melting point, there is color, and there is solubility. All these could be used to determine the purity of a particular substance in a mixture. But in this case, the first technique is paper chromatography. Which are the criteria in this case? Which are some of the properties we use in paper chromatography? We'll be using color, and we will be using solubility. In color, substances are made up of color except white. Except the white color, which represent purity. These colors can be separated into their pure forms using a paper chromatography technique. Then secondly, the ability of a substance to dissolve in a solvent will determine how it can be separated. For instance, in a mixture containing dyes, a mixture containing dye can be separated using a paper chromatography. What do I mean by that? I mean that the higher the solubility, the faster the color travels up the paper. Different colors have different solubilities. Straight away, what do you understand by a paper chromatography? Paper chromatography. This is an analytical technique or method that uses a paper to separate components in a mixture. It's an analytical technique used in chemistry to separate components in a mixture. Paper chromatography comprises of a paper, which is the stationary phase. It also has a solvent. It also has a solvent, which is the mobile phase. There is also the lid and a clamp to cover the beaker, in a case where the solvent is volatile. Usually, the ultimate goal of this technique is to determine the purity of the substance by investigating if there are some impurities present in the mixture. This technique is usually used to separate colors in dyes. It is usually used to separate colors in dyes. It is used in many other industries. beside the fact that it is used to separate colors in dyes, it is also used in many other industries in the 21st century. It is used in the pharmaceutical industry to detect impurities in new products. It is also used in the food industry to detect contaminants or impurities in foods and also in drinks. In the agriculture and wine producing companies, it is used to determine or for the study of ripening and fermentation. For law enforcement department, it is also used to detect drugs and dopes in animals and humans. For the cosmetic industry, it is used in the analysis of cosmetics. In research centers, it is used for the analysis of a reaction mixture in a biochemical laboratory. It is also used in the environment and the water supply companies. In the environmental sector and the water supply companies, it is used to determine the level of pollutants or the level of impurities in water supply. And above all, in the health sector, especially in hospitals, it is used to detect blood or to detect alcohol levels in a patient's bloodstream. So this technique is very, very important. Very, very important. How does a paper chromatography work? As I said initially, the paper chromatography contains, there are two phases in the paper chromatography. We have the stationary phase, which is the paper, and we have the mobile phase, which constitute the solvent and the mixture that has been dissolved in it. We call it the mobile phase because the solvent and the dyes or the components in it will move or will travel up the paper. That's why we call them the mobile phase. Now, as I said, a lead is also important in this case, especially when our components for or the analytes is a volatile liquid. In order to prevent it from evaporating, we need to cover it. So now, how does a paper chromatography works? The first thing in this is you need to draw. In a paper chromatography, you need to draw. Use a pencil to draw a line. You need a pencil to draw a line at the basement on the paper. We use but a pencil to draw a line here. We use the pencil to draw a line right here. On that line, that is where you can spot the dye. And the line, the pencil line must be above the liquid. It should not be in the liquid or in the solvent. It shouldn't be in the solvent. It should be above the solvent. On that pencil line, that's where you can spot different dyes for investigation. You can spot different dyes on this line for investigation. And taking, for example, this is a sample spot here on the line. The sample spot on the line, and which is above the solvent. After a period of time, what happens? The solvent will travel up. The solvent will travel up the solvent will travel up the paper. This is the direction of the flow of the solvent. It will travel up the paper. depending on the solubility of the component of the mixture. Depending on the solubility of the mixture in this sample, because I said the more the solubility, the more soluble the substance is, the faster it moves up, or the faster it travels up the paper. So since we said that different substances, they have different solubilities, some will dissolve first, some may dissolve after a period of time. So when they dissolve, when the mixture now dissolves in the solvent, what happens? They will travel up the paper. As they will be traveling, the different components in the mixture will be separated and will be indicated by the different colors. Here, this is what you see. After some time, what happens? The mixture now dissolves. After it dissolves into the different components. The different components based on their solubility. This is how they travel up the paper. As they are traveling up the paper, the colors are being separated. They are separated. So now, we can now determine the impurities in this compound or in this mixture. And we will determine it based on our RF value, the retention factor. We will measure the distance moved by this blue. We'll measure the distance moved by blue. We will also measure the distance moved by this green. We will also measure the distance moved by this red. And we'll also measure the distance moved by this yellow and this other purple here. Let's see the second example. Now, this is a standard that has been made. The mixture, this is the mixture. There are different dyes, dye 1, dye 2, dye 3. This is a pencil line that has been drawn, called the basement line, the pencil. Remember, I said it is recommended to use pencil, not pen, because if we use a pen contains many colors. The blue pen, apart from white, all other colors are a mixture of other colors. Blue is a mixture of other colors. Yellow is a mixture of other colors. Red is a mixture of other colors, apart from white. White signifies purity. But any other color or any other dye, it has a combination of other colors. So now, these are the dyes. These are the different dyes here. And this is a mixture. This is a standard here. The standard, they want to determine the type of dyes, whether the type of dyes here are the ones in this mixture here. We want to see which of the dyes has impurities in it that are present here. Have impurities in it that are not here. Or have the same components that are present in the mixture. Now, if we look at dye number 1, dye 2, dye 3. The mixture has two colors. There is blue and there is a red. Now, dye 1 shows a relationship between in the mixture. It means that dye 1 has a component that is present in the mixture, and they are at the same level. They are the same color and at the same level. They have traveled the same level up the paper. Dye 2 has only one color in it, and that one color is not present in the mixture. So dye 2 has no relation with the mixture. Dye 3 has only one color in it, and that color is also present in the mixture. So the only dye that has something that is not present in this mixture is dye 2, because it has yellow. and yellow is not present in the mixture that have been set as a standard. So the only two dyes that have something to do with the mixture, which is the standard, are dye 1 and dye 3. Dye 1 and dye 3 are the ones that have components that are present in the mixture. Now let's go to example number 3. Example number 3. We have soluble, water soluble dyes. These are water soluble dyes here. These are water soluble dyes. We have dye A, dye B, dye C, dye D, and dye E. These are water soluble dyes. We want to see dyes that are related, dyes that have relation with other dyes here. If we look critically, dye A has 2 colors. The colors are not present in dye B. The colors are present but in dye C. One of the colors is present in dye C, and one of the colors is present in dye C. 2 of the colors are present in dye C. The colors are not present in dye D. The colors are not present even in dye E. Therefore, dye A and dye C, they have something in common. They have some components in them that are present in each other, as seen on the as seen on the as seen here. Something here has something here to do with this. So that's why we say, I can put it like this. This one has something to do with this, and this. So I now say A and C have some components that are present in each other. Now, let's proceed to the other water soluble dyes C, D, and E. If I look at B, it has nothing in common with C, D, and E. Let's go to C. Dye C has yellow, which is only present in A. We have already mentioned that. It has A. It has red. And you look at the red in dye A, I mean dye C, it is also present in dye E. So we now say dye E has something here in common here. So dye C has something in common with dye E. Again, if we go up the paper, the components have traveled, many components have traveled up the paper. At this end here, you see that the blue, the deep blue, has something in common. The deep blue present in C is also present in E. So we now say they have something in common. So dye C again and dye E have some components that are present in each other. So an investigator will know the ones that are impurities in this and in all these water soluble dyes. In that case, B and D have substances. B and D have substances that are not present in A, C, and E. I said B and E. In that case, B, not E. B has a substance that is not present in A, is not present in C, is not present in D, and is not present in E. Is only what? Is only B. B is the only substance that have components that are not present in A, C, D, and E. Okay, here you can When given such an investigation, then if you want to determine the components that are present there, we need to use what we call the RF value. And what is the RF value? That is the retention factor. The retention factor, these are standard factors, these are standard values that have been laid down in data booklets. Every substance has an RF value. All these values are found in data booklets. When we have an impurity, we calculate the RF value. We go to the data booklet. We refer the value. We refer that value there and look what. And from there, we will know. We will know the component, or we will know that the identity of the impurity. How do you use the retention factor? RF, which is known as the retention factor, is used to determine the impurities in substances or in a mixture. This is how we calculate it. The RF value, or RF, is given as the distance traveled by the component divided by the distance traveled by the solvent. Okay, now this is an example of it. This is an example of it. We have, this is the base line, and which is the pencil line. Now, there was a mixture here. From this mixture, different components from this mixture, the mixture was dissolved in the solvent. After dissolving in solvent, the components had to travel up the paper. The distance traveled by the different components was dictated using a ruler. Now, the first component was red. This is the red color, has a red color. The second component has a purple color, and the third component had a blue color. Now, the distance traveled from the pencil line, or the baseline, for red was 2 centimeters. The distance traveled by the purple component is 6 centimeters, and the distance traveled by the component which is blue, that has a blue color, is 10 centimeters. Now, the total distance traveled by the solvent, of the paper is 10 centimeters. This is the 10 centimeters. 10 centimeters. So now, to know the RF value for each of these components, let's say the RF value for the component that has a red color will be what? The distance traveled by red, which is 2 centimeters, divided by the distance traveled by the solvent. The distance traveled by the solvent is 10 centimeters. So the RF value for red will be 2 centimeters divided by 10 centimeters. When we have the value, we go to the book of data where the RF values are there, and we refer. From there, we will know the component. We will know the component that has that value. Here, for F, let's say for F, this is it. For red, the RF value for red was 2 centimeters. Okay, 2 centimeters divided by 10 centimeters, which is the distance traveled by the solvent. And now, what will be the RF value? The RF value for the red component will be 0.2. We now go to our book of data. Under the RF value, we'll see under the RF values, we will see that component that has 0.2 value. That component, from there we will identify the component. We'll know the component, the impurity, the name of the impurity found in the mixture. That is how you do it. Okay. So we said that these values are used to identify the component of mixture. They are used to identify the components of mixture. So the RF value in the book of data will give you the identity of that component. the RF value of a particular compound is always the same. And this, it makes it easy for chemists to identify unknown substances or impurities in a mixture. Okay, learners, during an exam, you could be given about 8 marks to describe briefly, or to describe an experiment. In that case, take for instance, they want to test your knowledge on chromatography, paper chromatography, and they tell you, plan an experiment to separate the colored pigments present in a substance X. You have been given a substance X. They tell you, design an experiment to show how you can separate the different color pigments in it. They just want you to do the procedure that is needed in chromatography. In planning your experiment, what are your materials? The material needed there in the laboratory, you have an eye protection, a piece chromatography paper, 250 cm3 beaker. You need a glass rod. You need a sticky tape. You need water as your solvent. You need a pencil, and you need the substance S, which is the ink. Now, what is the method and the procedure used in this thing? In your experiment, outline it: material, method, and procedure. On your procedure, outline clearly the stages. 1. Use a pencil to draw a line at the basement of a paper chromatography. 2. Spot of the pigment is placed on the middle of the middle of the drawn line of the paper and allowed to dry. The paper is placed in a suitable solvent, for example, water that we used, in a beaker. Remember, if the solvent is volatile or it vaporizes easily, it is advisable to put a lid or a cover on the beaker to prevent the vapor from escaping. Again, as the solvent rises or travels up the chromatography paper, the dyes in the ink or in the mixture will separate. I've outlined it here. This is the mixture. This is the mixture X. Let's say this is the mixture X. Now, the mixture X, as it dissolves in the solvent, what happens? The components there will do what? As they travel up the chromatography paper, they are being separated. The distances are taken. The distance of each component is being taken. The final distance, that is the solvent front, the distance traveled by the solvent, is also taken. Now, we now go to our RF value. How do we calculate our RF value, our retention factor value? I said the formula is distance moved by component divided by distance traveled by the solvent. And we came here, we have done what? We have resolved them here. For red, our red, which I did here, I took the red here, the first red here, the red here, the first red there was 2 centimeters. It traveled 2 centimeters, and the RF value was 0.2. The second one was purple here, and the RF value is 0.6. The third one was blue. It traveled 8 centimeters, and 8 centimeters divided by 10 centimeters, the RF value was what? The RF value was 0.8. Then the next step was we now go to our book of data. The components in the mixture can be identified by comparing their RF values with the RF values in the data book. So when we compare with the other value in the data book, we will now spot out what is the component F, the identity of the component F, the identity of the component purple, and the identity of the component that has a blue color. Conclusion in your experiment. Your conclusion should be that the different spots or the different color is an indication that the component has impurities in it. The substance X has impurities in it, as indicated by the different spots, the different colors. If it was only one spot, we would have said the component or the substance X is pure. But because it has different colors in it, we say it is an indication that it has impurities in it. So now that is how you can plan an experiment to use the paper chromatography technique to detect impurities or the different component in a mixture. Now we are going to start with activity 2 to see how you have better understood the technique. This is a past paper, part of a past paper. The past paper reads: chromatography is used to separate a mixture of colored dyes. Three different dyes or three different dye mixture, A, B, and C, were spotted onto a piece of chromatography paper. Two pure dyes, X and Y, were also spotted onto the sample piece of paper. The diagram below shows the result of the chromatography. That is the diagram. It shows the solvent front on the basement line, and it shows the different dyes and the pure dyes, which are X and Y. Okay, question A. State the name of a piece of apparatus that could be used to spot the dyes onto the paper. What apparatus could be used to spot the dye on this spot, to spot the dye on the paper, on this baseline? What apparatus was used to spot the dye on A, to spot the dye on B, to spot the dye on C? In that case, you must use a very small or tiny capillary tube. It must be a small or tiny capillary tube. For you must use a small, tiny capillary tube to spot the dye onto the paper. Question number 2, B. Suggest why the baseline was drawn in pencil, not in ink. Now, drawn in pencil and not in ink. Here, we can say draw in pencil and not in ink. We can say, as I said, if we use ink, ink is a dye. Ink has many colors present in it. If we use ink, it could be blue, it could be red, it could be yellow. They have a mixture of other colors. So ink will dissolve, and when ink dissolves, ink will undergo chromatography. When we use but ink to draw the line there, the ink will dissolve, and when the ink dissolves, it's going to undergo chromatography. The components there are going to travel up the paper. So ink is not good. Ink is not good. A pencil is preferable because the pencil will not dissolve. Once more, ink will dissolve, ink will travel up the chromatography paper, and it will interrupt the result. But pencil will not dissolve. C. Which dye mixture contains both dye X and dye Y? If you look at it, which of the dye mixture contain Which dye mixture contains both X and Y? This is X. If we look at it, X here, this is the spot here. This X here. So this is X here. That is X. X is present in B. Y is here. That's good. So we now say C is B. Which dye mixture contains both dye X and dye Y? C is B. All this dye here in B is present in this dye here in B is present in X, and this dye here in B is also present in Y. So our answer is C. Our answer is B. Our answer for C is B. Question D. Which dye mixture does not contain X or dye Y? Which of the dye mixture does not contain X or Y? Okay. Look critically. B. B contains, yep. For question number D, which dye mixture does not contain X or dye Y? Okay. Does not contain. You will see that question number D, B, we cannot take B because B has, we cannot take C because C has some components there. C has some components that are related to Y. Now let's take A. A, here, there's no X or Y. Here, there's no X or Y there. Here, there's no X or Y there. Okay, here for A, there's no X or Y. Then here, there's no X or Y. There's no X or Y. Here, there is no X or Y. There's only here. So there's no X and there's no Y. So our answer for D is A. Our answer for D is A. Which dye mixture does not contain dye X or dye Y? Our answer is A. E. E. In which mixture A, B, or C has the greatest number of dyes? In which mixture A, B, or C has the greatest number of dyes been separated? Which of these mixture has the greatest number of dyes that have been separated? Okay, now number of dyes that have been separated, we have 1, 2, 3, 4, 5. B has 1, 2, 3. A has 1, 2, 3, 4. So our answer for E, A, B, or C has the greatest number of dyes been separated. We have, how many dyes have been separated? 1, 2, 3, 4, 5. There are 5 of them components that have been separated. So C, mixture C is the right answer for E. So E is C. It drives us to activity 3. Activity 3 is another past paper, a part of the past paper. A student investigated the colors. A student investigated the colors present in a fruit drink. The fruit drink was tested to check that no artificial colors has been added. The apparatus below was used. This is the apparatus below. You have the paper, you have the solvent, you have the beaker. and you have the glass cover. The glass cover, as I said the other time, in case there is a volatile solvent, to cover it from evaporating. So name the method used. Which method or which technique is this one? The technique here, the separation technique used here is what we call paper chromatography technique. This is a paper chromatography technique. Paper chromatography technique. 2. Sub 2. Why is there a glass cover on the beaker? As I said, either you use a glass cover or you use a lid, any lid. Here it is a glass cover. The glass cover is to prevent the solvent from evaporating. Definitely, the solvent present here is volatile. So it's to prevent the solvent from evaporating. Question B. When should the paper be removed from the beaker? When should this paper here be removed from the beaker? I said the paper can be removed when no more color is observed, or when no more color is traveling up the paper. When no more color is observed here, when no more color is observed traveling up the paper, or if no color is observed at all traveling up, it means all the colors, all the mixtures have been dissolved, all the colors have been separated. So if there's no color traveling up again, we can now remove the paper. So conclusively, we say it is removed when no more color is observed or no more color is traveling up the paper. It takes us to question C part of it. The C part of it. The C part of it. This is the C part of it. The diagram shows the result of the experiment. We have the chromatography paper there well outlined. The solvent front. This is the distance traveled by the solvent. This is the color from the fruit drink. This is the fruit drink here. The color. These are the color mixtures that have been separated from the fruit drink. Then these are other artificial colors. These are other artificial colors. These are other artificial colors that have been separated. The question reads: how many different colored compounds were present in the fruit drink? How many different colored compounds were present in the fruit drink? In the fruit drink, we have, these are the compounds there. This one, in the fruit drink, we have 1, we have 2, we have 3, and we have 4. That is it. Those are the ones going up. Now, these are artificial colors. Now, from the artificial colors, we can identify. From the artificial color, there is one artificial color that is present in the fruit drink. This is it here. There is one artificial color that travels up. There is another one that also travels up. There is another one that also travels up. So there is one artificial color that has been identified that is present in the fruit drink. They have traveled the same distance. That's why they are at the same spot. They have traveled the same distance from the basement. So we say this one is present in the fruit drink. Now, let's check if there is another element. If there's another one there. So, how many different colored compounds? How many different colored compounds were present in the fruit drink? Okay. From the artificial compound, This one is present in the fruit drink. So the only two that were not in the fruit drink are what? This one here, and this other one here. So there are two different colored compounds that were present in the fruit drink, because this one is already there as identified by the chromatography paper. So there are only two. So there are two. We say there are two different colored compounds present in the fruit drink here. So two artificial colors. So C1, how many different colored compounds were present in the fruit drink? We have two. Okay, C2, are there any of the artificial colors present in the fruit drink? Explain your answer. I have already explained it above here. Yes. Are they any of the artificial colors present in the fruit drink? Yes. The reason is this, because one of the colors soon have traveled up the same distance with that in the fruit drink. One of the artificial color have traveled up the same distance with that in the fruit drink. Or we can say one of the artificial colors have the same retention factor value with one of the artificial colors have the same RF value with another one in the fruit drink. Why do I say so? Because if we calculate the distance that this one here has traveled and the one that this one has traveled, since they have traveled the same distance, they will have the same RF value. So we can say yes, because one of the colors sun have traveled up the same distance with that sun in the fruit drink. Or we can say yes, because one of the colors have the same RF value with another one in the fruit drink. That's simple. So fellow learners, this is just an example. You will have similar questions like this. So go solve many other questions from alternative to practicals exam. You will see many other similar questions there. Remember, you should know how you can obtain your RF value. You should also know the value, the purpose of the RF value. You should also know how you can plan an experiment to separate pigments present in any substance you are given. You should know that the different components or the different impurities present in the mixture are identified by their RF value, and this is through the distance that they have traveled up the chromatography paper. If you are asked to define RF, what is RF? What is RF? Or what is retention factor? What do you understand by retention factor? I think the better thing to do is write by this expression. RF is equal to distance traveled by component divided by distance traveled by solvent. If you are asked to define it, please advisable to give by this expression. You should, when you are given a question, learners, you should study the question. You should study the diagram. What is in the diagram? Try to draw the relationship in the diagram before going to answer your question, before answering your paper. Take the paper, try to see the distance traveled by the different components, then try to see the relationship between the different dyes, and try to draw out your conclusion. Then you start answering your paper, start answering your question. Thank you. Thank you for listening. I advise you, try to share share the video, like the video, and subscribe so that you will be observing many other laboratory videos. Thank you once more.